Welcome to Gnana Deeksha, concept-based digital learning. I am Dr. Sumathi Natarajan, lecturer in microbiology at Telangana Social Welfare Residential Degree College for Women, Mahendrahills. In today's session on immunology, we will learn about antigen and antibody reaction. Any foreign particle entering our body is called an antigen. We fight against these antigens by producing antibodies. Antibodies are constantly interacting with antigens in our body even as we speak now. A basic knowledge of these reactions is important to understand the principle and working of numerous diagnostic tests and also various immunoreactions occurring in our body. Today we will learn how an antigen and antibody interact. What are the different forces that play a role in antigen-antibody reaction and the various factors that influence these reactions. We will also look into the four main types of antigen-antibody reactions, which are precipitation, agglutination, neutralization and complement fixation. Finally, in vitro applications of these reactions will be covered. Before we start off, let's understand the concept of antigen-antibody interaction by taking a simple example. In this figure, from all the different types of keys surrounding this lock, only one key can open the lock. So, a key is specific for a lock. In a similar manner, an antibody is specific for an antigen. If we look here, the structure of an antibody is Y-shaped. The part of the antibody that interacts with an antigen is called fragment of antigen binding or FAB fragment. The lower part of the antibody is called as fragment of crystallization or FC fragment, which interacts with cell receptors and is involved in immune response and signaling. The variable regions of the FAB fragment that interact and bind to an antigen is called paratope. The site on an antigen which triggers an immune response and where an antibody binds specifically is called an epitope or antigenic determinant. Simply, antigen-antibody reactions involve interaction between an epitope on an antigen and paratope on antibody. Different epitopes can elicit different antibody response. So, antibodies show specificity in diversity. Here we see an antigen like bacteria possess various epitopes on their surface. Each epitope can elicit a specific antibody response. Many epitopes are present on bacterial cell surface. If two copies of same epitope are present, then it is called a multivalent antigen. In the second image, we see an antigen with different epitopes binding to different types of antibodies. Such an antigen is called a polyvalent antigen. All antibodies are immunoglobulin. We are all aware of the 5 different classes of immunoglobulins. They are immunoglobulin G, A, M, E and D. We also know that immunoglobulin A can exist as a dimer and immunoglobulin M as a pentamer, whereas immunoglobulin G, E and D exist only as monomers. Here, antibody valency means the number of epitopes an antibody can bind to. So, any antibody will have a minimum valency of 2. Whereas, immunoglobulin A has a valency of 4 and immunoglobulin M a valency of 10. Now, let's get an idea of the forces that play a role in antigen-antibody reactions. When an antibody and an antigen interact, it is by means of weak non-covalent bonds like hydrogen bond, ionic bond, hydrophobic interactions and van der Waals forces. Antigen-antibody reactions are weak and non-covalent forces operate between them. So, they are reversible. Let us have a look at various factors that influence antigen-antibody reaction. These include affinity, avidity, cross-reactivity, and the concentration of antigen and antibody, also called as the antigen-antibody ratio. Let's understand these factors one by one with an example. Affinity refers to how strongly a single epitope and a paratope are bound, whereas avidity is the overall binding strength of multiple epitopes and paratopes on a single antigen. Since immunoglobulin M exists as a pentamer, it has high avidity. However, immunoglobulin G is known for its high affinity. To understand this concept, let's consider an example. Here, if we imagine the swing as an antigen and the child as an antibody, how strongly the child holds on to the swing is an example of affinity which we can see in the first image. In the second image, we see many children are holding on to a single swing. The overall strength with which the kids are clinging on to the swing is an example of avidity. Another factor influencing antigen-antibody reactions is cross-reactivity. Now, although antigen-antibody reactions are very specific, sometimes an antibody elicited by one antigen can cross-react with another antigen. This can happen when two different antigens share identical epitope or structurally similar epitope. A simple example of cross-reactivity comes from our history where Edward Jenner used cowpox virus for immunization against smallpox. This was successful due to cross-reactivity of antibodies. One more crucial factor is antigen to antibody ratio. Either excess of antigen or antibody will not result in appropriate antigen-antibody interaction. It is always desirable to have an optimal antigen-antibody ratio for antigen-antibody complex formation at the zone of equivalence. Complex formation does not occur if antibodies are in excess, known as the prozone effect, or when antigens are in excess, called as the post-zone effect. With these concepts in mind, we are now ready to learn different types of antigen-antibody reactions. There are four main types of antigen-antibody reactions: precipitation, agglutination, neutralization, and complement fixation. Before we proceed further, let us understand what are quantitative and qualitative tests. A quantitative test tells us if a desired molecule is present or not, whereas quantitative tests can give us number of molecules that are present. For example, looking at this image, if I ask you whether there is any green candy, you would say yes, this is a qualitative test. Now, when I ask you how many green candies are there, you will reply with a number like 2. This is a quantitative test. Let us start with precipitation reactions. When an antibody reacts with a soluble antigen under optimal antigen-antibody ratio, it will result in the formation of an insoluble lattice visible as a precipitate. The reacting antibody is called a precipitin and participating antigen is termed precipitinogen. Two main conditions must be met for precipitation reactions to occur. First, antibody must be polyclonal. Polyclonal here means ability to bind to multiple epitopes on an antigen. Second, apart from being soluble, an antigen must be bivalent or polyvalent. This means it must have at least 2 copies of the same epitope or have different epitopes that react with different antibodies. Precipitation reactions are performed in vitro, in fluids or on gels. For observing precipitation reactions in fluids, a fixed volume of antibody solution is taken in a series of test tubes to which increasing concentration of antigen is added. In tubes with optimal concentration of antigen and antibody, a visible precipitation is observed. This is due to an insoluble lattice formation that occurs at the zone of equivalence. As seen in the image, plotting the amount of precipitate against increasing antigen concentration yields a precipitin curve. These precipitation reactions find use in diagnostics, where antiserum is tested by overlaying it with antigen solution. Here we test if serum from patient contains optimal antibodies towards the antigen to confirm infection. A visible precipitin ring indicates a positive test. Precipitin ring test is used in diagnosis of anthrax and also for classification of streptococci by Lancefield method. Precipitation reactions can also be carried out on gels called immunodiffusion to give a visible precipitin line at the zone of equivalence. There are 2 types of immunodiffusion reactions. Radial immunodiffusion, also called as Mancini's method, and double immunodiffusion, commonly known as Ochterlony's double diffusion. Immunoelectrophoresis is an other technique that combines electrophoresis with double immunodiffusion. Coming to radial immunodiffusion, it is a quantitative technique where agar containing specific antibodies is poured onto a petri dish or glass slide. Wells are then created in which antigen solution is added. As the antigen diffuses into the agar, it forms a precipitin ring with the antibodies at the zone of equivalence. Area of the precipitin ring is proportional to the concentration of antigen. By comparing area of precipitation ring with a standard curve, the concentration of antigen in the sample can be determined. Double immunodiffusion is used to test similarity between antigens. Ochterlony's double immunodiffusion involves placing the antigen solution in different wells on an agar slide with antisera is placed in a central well. Upon incubation, antigen and antibody diffuse and at zone of equivalence, precipitin lines are formed. Different patterns of precipitin lines tell us about the epitope similarity among the antigens. First one, as we can see, is the image where 2 precipitin lines are crossed. Shows 2 separate reactions, meaning antigens are unrelated and share no common epitopes. This is called non-identity. The Y-shaped second reaction shows a continuous line with a spur at one end, indicating a partial identity of epitopes on antigens. A third reaction shows a precipitin arc formation, indicating a full identity and the presence of common epitopes among the antigens. Since immunodiffusion is a slow process taking nearly 12 to 14 hours until the presbyopia lines are visible, a new technique called immunoelectrophoresis works faster as it combines electrophoresis with immunodiffusion. In immunoelectrophoresis, the antigen mixture is first electrophoresed on agarose gel. Troughs are then cut in gel parallel to the direction of electric field. Antiserum is added to the trough. Antibody and antigen diffuse towards each other and produce lines of precipitation where they meet at optimal ratio. This technique is used in clinical laboratories to detect the presence or absence of proteins in the serum. It is useful in detecting immunodeficiency diseases. For example, in the last image, when we compare the immunoelectrophoretic pattern of normal serum in top half with that of the patient's serum at the bottom half, we see a precipitin arc is missing in the immunoglobulin A region, indicating an immunoglobulin A deficiency. Immunoelectrophoresis is a strictly qualitative technique. A related quantitative technique is rocket electrophoresis, which measures antigen levels. In this technique, a negatively charged antigen is electrophoresed in a gel containing antibody. The precipitate formed between antigen and antibody has the shape of a rocket, the height of which is proportional to the concentration of antigen. With the help of a standard graph, the concentration of antigen in the sample can then be quantitated. Another type of antigen-antibody interaction is agglutination reaction. While precipitation reactions involve soluble antigens, agglutination reactions involve particulate antigens. Particulate antigens include red blood cells and also microorganisms like bacteria, fungi, and protozoa. Cross-linking of different epitopes present on particulate antigens by polyclonal antibodies results in visible clumping called agglutination. Antibodies involved in agglutination reactions are called agglutinins and antigens are called agglutinogens. There are two main types of agglutination reactions: direct or active agglutination and indirect or passive agglutination. Active agglutination occurs when optimal ratio of antibodies bind to particulate antigens resulting in visible clumping. Active agglutination is used in diagnosis. For example, in blood grouping to detect antigens on our red blood cells. For the diagnosis of typhoid by performing Vidal test. And diagnosis of various other diseases like brucellosis, typhus, leptospirosis where antigens in blood are detected. Since antibodies can agglutinate bacteria, agglutination reactions are used for serotyping of bacteria based on various epitopes present on bacterial pathogens. Active agglutination reactions are used in blood typing. The presence of different antigens on the surface of RBCs and the presence or absence of antibodies in serum are used for the grouping of blood mainly into four groups: A, B, AB and O. Presence or absence of RH or D antigen determines positive or negative blood group. Blood grouping of a person is determined by taking a white tile and mixing 3 different drops of blood with antisera A, antisera B, antisera D. It is then observed for any visible agglutination which indicates a positive reaction. In the table, we can see different antibodies present in plasma of different blood groups. The second image represents agglutination reactions of various blood groups. The image on the right, we can see visible agglutination occur with antisera A and antisera D, indicating blood group as A positive. Prior to blood transfusion, it is vital that donor's blood and receiver's blood is well matched or compatible so that no fatal reactions occur in the body due to agglutinations. Therefore, before a blood transfusion, a drop of receiver's blood and donor blood of same group is mixed and observed under microscope for agglutination. If there is no agglutination, then it is safe to transfuse the blood. The sensitivity and simplicity of agglutination reactions can also be extended to soluble antigens by the technique of passive agglutination. The soluble antigens are made particulate by coating them onto carrier particles like RBCs, latex beads, polystyrene and bentonite. When antigen-coated beads are allowed to react with antibodies, visible clumping results. These reactions are used in diagnosis of bacterial and viral diseases to detect rheumatoid factor and also in pregnancy test kits to detect HCG. Let us now learn about another antigen-antibody reaction called neutralization reaction. Since antibodies are specific and bind to anything foreign, they can protect the cells by blocking foreign invaders. What I mean here is antibodies can bind to viruses or foreign proteins like bacterial exotoxins, not allowing them to bind to and show their effect on host cells. This property of the antibody that neutralizes the exotoxin and inactivates the viruses is the basis for neutralization reactions. Antibodies that bind to bacterial toxins and neutralize their harmful effects are called antitoxins. We can see here that toxins can cause cell damage, but when neutralizing antibodies are present, the harmful effect of toxins are neutralized. Thus, they protect our body from action of various exotoxins like diphtheria toxin and tetanus toxin. Neutralization reactions are also used in diagnosis of certain viral diseases. Some viruses like influenza, measles and mumps virus can bind to sialic acid receptors on the surface of RBCs with the help of their hemagglutinin spikes. This binding results in visible clumping, which is called as hemagglutination. If neutralizing antibodies against the virus are present, then the viruses will be neutralized by these antibodies so that they cannot bind to RBCs and there will be no hemagglutination. This is called hemagglutination inhibition assay, which is used to estimate the antibody titer against various hemagglutinating viruses. Now, let's look into complement fixation reactions. Complements are inactive serum proteins that can be activated by either antigen-antibody complexes or bacteria and bacterial products. Once activated, complement proteins initiate a cascade of proteolytic events leading to the formation of a membrane attack complex that ultimately results in elimination and direct lysis of the infecting agent. Let's now see how complement fixation reactions are used as a diagnostic test for detecting the presence of antibodies against specific microbial infections. CFT or complement fixation test is based on the principle that antigen-antibody complex fixes complement. We all know that serum contains antibodies. The first step in the complement fixation test. We all know that serum contains antibodies. The first step in the complement fixation test involves addition of known antigen to a patient's serum. This is then followed by the addition of complement to the solution. If the serum contains specific antibodies, the complement will be activated and fixed by the antigen-antibody complex. However, if there is no antibody in the patient's serum, the antigen-antibody complex will not be formed. So, complement will not be fixed but will remain free. As coupling of complement to antigen-antibody complex has no visible effects or changes, it is necessary to use an indicator system. This consists of sheep RBC and anti-sheep RBC antibody to check for free complement from the previous step. So, the second step detects whether complement has been utilized in the first step or not. This is done by adding the indicator system. If the complement is fixed in the first step due to the presence of antibody, there will be no free complement to fix the indicator system. However, if there is no antibody in the patient's serum, complement will be present free and can now react with antibody-coated sheep red blood cells to bring about their lysis. No lysis of sheep RBCs indicates the presence of antibody in the test serum and is a positive CFT, while lysis of sheep RBCs indicates the absence of antibody in the serum and is a negative CFT. That's about complement fixation test, which is used to screen a large number of viral and bacterial infections. Till now, we have looked into various antigen-antibody reactions. These are mainly used in disease diagnosis and to determine relative concentrations of antigen or antibody. Let us now see some of their applications. Major application is in diagnosis to detect various diseases such as syphilis, typhoid, hepatitis and HIV. These reactions are also used to confirm presence of antibodies in the serum. As we have seen, agglutination reactions are used for blood grouping and blood matching prior to transfusion. These reactions also find use in forensics for identification of blood and seminal stains. Antigen-antibody reactions form the basis of various amino acids such as ELISA, RIA, Western blotting, which are used to detect various proteins. During clinical trials in vaccine development, to know the efficacy of a vaccine, blood samples from clinical trial volunteers are checked for presence of antibodies and know the antibody titer using such antigen-antibody reactions. As seen previously, various immunodeficiencies can also be detected by antigen-antibody based reactions. In the current situation, it won't be appropriate if we do not discuss the diagnosis of COVID-19, which is also based on antigen-antibody reactions. The kit uses the principle of immunochromatography, which is the separation of components in a mixture through a medium by capillary force with specific and rapid binding of an antibody to its antigen. Each cassette is a dry medium that has been coated separately with anti-human IgM and IgG at test lines and anti-rabbit IgG at control line. The cassette contains a sample pad and a conjugate pad. In the sample pad, sample such as blood or serum is added for detection of IgM or IgG antibodies against coronavirus. The conjugate pad contains colloidal gold labeled rabbit IgG and gold conjugated novel coronavirus antigen. Once diluted serum or whole blood is applied to the sample pad, if IgM and IgG against COVID-19 are present in the sample, these antibodies will bind to gold conjugated coronavirus antigen, forming antigen-antibody complex in the conjugation pad. The immune complex will then move across the cassette medium via capillary action. The immune complexes will be bound by anti-human IgG or IgM at the test line and develop color indicating a positive test. If there is no coronavirus IgM or IgG in the sample, no binding will occur at the test line and no color will develop indicating a negative test. In any case, the gold labeled rabbit IgG will bind to anti-rabbit IgG antibody at the control line. These control lines should show color as this confirms that the kit is working properly. As explained, color should develop in control line or C line indicating that the kit is working properly. If only G test line develops color, it indicates a previous infection with novel coronavirus. If only M test line develops color, it indicates a recent infection with COVID-19. Color at both G and M line also indicates recent infection with COVID-19. No color at both G and M line indicates no infection or antibodies are not present at detectable levels in the blood or serum. Unlike detecting antibodies against coronavirus, there are also other kits available which detect novel coronavirus antigen in nasal swabs. Let us revise what we learned today. We are aware that antibodies exhibit specificity in diversity. Paratopes are regions on antibodies that interact with epitopes on antigens. This binding between a paratope and epitope forms the basis of various antigen-antibody reactions such as precipitation, agglutination, neutralization, and complement fixation. Precipitation reactions involve soluble antigens, whereas agglutination reactions involve particulate antigens. Blood typing and diagnostic tests for various infections are based on agglutination reactions. Neutralization reactions involve antibodies which neutralize harmful effects of bacterial pathogens and their toxins. Serum proteins, called complements, increase the effector response of antibody bound to an antigen, which forms the basis for complement fixation tests. Finally, we discussed the applications of antigen-antibody reactions and also their use in COVID-19 rapid test. This concludes our session for today. Thank you all and have a pleasant day.